Okay, thank you very much for having me here talking about this work that we've been doing in the past 2-3 years. And my name is Marco Antoniotti. I come from Milan. I've been here in Berkeley many years ago. But that's what I'm doing now. I'm going to talk about this set of algorithms that we have developed that attack the problem of inferring a progression model for cancer. And the motivation that spurred us to work on this problem was a collaboration that we started with some researchers on colon cancer in Milan at the Istituto Nazionale dei Tumori. And eventually we found all the work by Vogelstein where there is a initial, probably the first progression model was described in the paper. Now, as we went on, we, of course, bumped into other problems, and there are the problems of what happens when you have cancer that evolves and subdivides in, becomes more heterogeneous, and where you have different clonal populations that occupy the whole volume of the cancer. And this is a nice slide that I put up because this is an original drawing by Darwin. So just to make the whole thing interesting. Now, the problem that we started looking at was, suppose that we want to describe what a process of accumulation of different mutations, different alterations looks like. What kind of data do we have around? And it turns out that most of the data that is around is cross-sectional data. And cross-sectional data, this is an example. This is the Picnlc paper on lung cancer. And it always looks like this. You have basically different genes that have a certain alteration associated to it, and the columns are patients. Okay? So this is the kind of data that you get, and this is the kind of data that you have, for example, in TCGA. So there is very little timing information associated to this kind of data, and our problem is to reconstruct, to superimpose a temporal structure on this sort of this data, to extract a temporal structure from this data. So turns out that there are two ways of looking at this problem. One way is what we call the ensemble cross-sectional data, which is what can be done on data sets coming, for example, from TCGA. Okay? These are data that have a list of mutations, a list of alterations associated to each patient. And then you can do also what is called individual level analysis. And this is becoming more interesting and more important because of, for example, of single cell data or of data that is coming from sections of a single tumor. So, but on the bulk of our work has been on cross-sectional data. Okay, so the data is collected at time of biopsies, and that's basically the kind of the timestamp, the only timestamp that you have. Okay, so let's keep going, and here you have a summary of the two different look at the two different aspects that of the same problem. And I'm reporting this picture here because the algorithms that we developed can be applied in both settings, although on the individual level settings we have very little results to show. We just looked at the Gerlinger's data set, which was mentioned in one of the previous talks, but for the time being it's we are writing up and we are trying to get more results out of it. So the main work that we've done is on ensemble reconstruction, and the ensemble reconstruction that we came up with is based on a particular, it's basically a Bayesian network learning scheme but it's based on a particular kind of score. And the score that we are using is, first of all, let me just tell you that there are several algorithms. Many of them have been originally developed here in Berkeley, if I remember. And of course, some of the people sitting in the audience have been doing a lot of work on this. Okay, all algorithms need some measure to decide whether and how to include an arc. And our choice is rooted in probabilistic causality theory. We are using a score which is derived from the theory of probabilistic causality that was developed by Suppis in Stanford over the years. He passed away about a year ago, if I remember correctly. It's a very simple idea up to a point. We reinterpret this scoring and the relation that it generates between different alterations as a relation of selective advantage between two alterations. Basically, we say that we can score the relation and we can say that one alteration confers the tumor a selective advantage in order to acquire subsequently the next alteration. This is an interesting reinterpretation which was suggested by one of the reviewers of one of the papers that we published on the subject, and it's also interesting because it kind of, let me just be very candid here, it makes the whole notion of causality much more palatable to biologists. This is biologist, maybe. Okay. In any case, but the bottom line is that the choice of this particular scoring method allows us to pack more information in the reconstruction algorithm. Okay, and the result that we get is actually not only a temporal without timing. Let's be clear. Okay, we haven't done work on timing yet. It's in the pipeline. But the result is that you can say that there is some deep relation between two events in one of the models that we come up with. So this is the typical example that we show. There is an assumption in our model is that all alterations are persistent, which opens up a can of worms regarding other kind of alterations that you may want to look at. You have in this case, for example, EGFR mutation gets to this cell keeps dividing, but because it has EGFR as a mutation acquired, then it has higher chance of getting a mutation on CDK, and therefore you get later on a clonal expansion of cells that have both mutations. Okay, so this is the simple example. These are the kind of probabilities that we need to compute, and the probabilities that we are computing are of two kinds. Well, actually, there is one specific kind, which is the condition that dictates what it means for one event to be the cause for another. It's a very simple conditional probability. And what is happening here is that you are just looking up, you're just observing the frequencies, and based on the frequencies, you are computing this probability. There is an extra little complication in here, in the sense that our setup is capable of dealing with potentially arbitrarily complex Boolean formula of combination of different alterations that have been acquired by a certain population of cells. Okay. This is a typical example. We have seen it somewhere in another talk before as well. It's the problem of deciding whether you have what happens when you have an overlap. You have patient A has these alterations here, patient B, sorry, you see this alteration here, you see this alteration B in these other samples, and you have an overlap. So the question is, what kind of network can you infer given these measurements? And this is wrong, because it would look like that from C you would infer A and leave B by itself. But if you want to look at hard exclusivity or even soft exclusivity, so standard OR or XOR, then these are the kind of networks that you want to infer. And our algorithms are capable of dealing with exactly with this problem here. Okay. So let me just give you a little bit of advertisement. This is the library. Okay. These are the 3 papers that we have published on the algorithms that are present in the library right here. These are the performance comparisons of our algorithms, CAPRI and Caprese. The difference is that Caprese reconstructs 3 topologies, while CAPRI reconstructs much more complex DAG topologies. Okay, and we have different kind of setups. These are data from this is simulated data, and these are some of the other tools that we have compared it against. And what you can see here is that, of course, well, one thing that you can see here, if you go a little bit more into details, is that our algorithms are also very robust with respect to noise. So the last thing that we've been doing recently is to have this set up, ensemble level, individual level, but above all, we have put together a pipeline for applying these tools. And these tools, CAPRI especially, is actually used at the very bottom, at the very downstream of a pipeline of analysis. And we decided, we did quite a bit of work. Actually, you will see later on who actually did the work, not me. In any case, we had to do a lot of work in order to analyze a specific data set. We have several data sets that we are analyzing now, but the main data set that we analyzed is the TCGA colon cancer data set. And the key point was that we found that in order to produce meaningful reconstructed progression models, you have to be very careful about what is the input data, and especially you want to be very sure that what you are putting into the system is corresponds to a very well-defined cancer subtype. And so a big chunk of the pipeline up to this point is to find cancer subtypes, driver events and cancer subtypes, and then going here. We are also using exclusivity finding tools in order to automatically produce some of the patterns, some of the formulae that we are then inputting into CAPRI. Okay. So this is one example of the result that you get out of CAPRI. This is TCGA code read. Okay. The interesting thing is that okay, this is work that's done in collaboration with the Oncologia Hospital in a research center in Barcelona. So this has been validated ex post by an oncologist. And the interesting thing is that we were able to find a role for, well, we were able to propose. We haven't validated yet experimentally, but we were able to propose a role for SOX9 as a downstream event of mutation acquired on ARID1A. Okay. So this is an interesting little result that came out of this analysis. So the TRONCO is the name of the algorithm suite. Is on Bioconductor. You can download it and play with it. And here are the main characters. Giulio Caravagna was the postdoc. Daniele Ramazzotti is finishing in 3 weeks and then is coming here to Stanford. And Alex Graudenzi, the other postdoc from CNR. And these are all the other people involved. He's a master student. There's Bud Mishra from New York University, who actually came up with the idea of using that particular score. Okay. Rebecca Sanz Pamplona and Victor Moreno in Barcelona worked with our setup. So final acknowledgments. We have Regione Lombardia especially gave some money for all of this. Okay. And some advertisement. I'm organizing a yearly school on Incomo. This year is May 24, 27. Okay. And you can check it out, and obviously you can come, and if you don't trust me, talk to Nico, who was there. I'm done.
Questions? I guess I can. Oh, go ahead.
I didn't quite understand the connection to the selective advantage, because there seems to be no notion of growth rate or expected number of offspring, also the typical.
Yeah.
That's the way to define fitness typically. So I'm wondering how you connect that and how you see that.
It's really not. We really do not have any particular interpretation in that sense. It's a selective advantage for us. Simply means that if you are looking at the data and you are really seeing a lot of alterations, okay, in one gene, then you can mathematically say that this population has a higher chance of acquiring a mutation later on. And so in some sense, your comment is that the way I interpret it is that maybe we are putting too much, we are going too much in the other direction. But seriously, this when in the first paper that we wrote, everything was described in terms of causality theory, and that really drew a lot of fire. And this was the suggestion about. So the term causality is really something, and it makes sense because. Well, you can go and talk about. You can go and look at the works from Judea Pearl, where you have this notion of intervention, and so how do you define an intervention by a mutation? Okay, so it's really we decided to tread very lightly and adopt a more neutral terminology, which would be more understandable by biologists. Yes.
Just to be, I don't think selective advantage has a neutral connotation. It's
what?
Selective advantage has a very specific connotation, and so I think maybe you've gone from one controversial
to another. Okay, I agree. Okay. I'll accept the criticism. Yes. Okay, and I'll keep it in mind. Yeah.
Okay.
Yeah.
Maybe the last question.
In the example of A, B, and C, in the mutual exclusivity example, so how do you know it's A and B causing C, not the other way around?
A and B, it's A XOR B or A or B. No, what I'm saying.
The other way around, like C causes.
Okay, that's yes. You could have the data may support also the opposite. Okay, but the data may support the opposite, but what we are doing, we are imposing some constraint on the way our causal implications are built. So the system will only find these sort of relationships. This is because, first of all, we are kind of cheating because we are inputting these nodes as atomic nodes in the seed network that then is used to do the structural reconstruction of the Bayesian model. Because as soon as you have a formula of this kind, and especially as soon as you have these junctions, your complexity blows up very, very quickly. Okay, so we are, in a sense, we are cheating, but it's not really being dishonest about it, in the sense that these, especially the exclusivity relationships, we either have them suggested directly by a clinician or a biologist, and this is the case in another study that we did on ACML. There are colleagues from the hospital in Milan that suggested two specific exclusivity patterns, or in the pipeline that I showed at the very end here. In this pipeline, the exclusivity groups are suggested by software like Mutex. Okay. So we are just taking this formula and we are just augmenting the input. We are just technically determined. We are lifting the input matrix in order to get these new nodes in the input.
Okay, that's great. I think we have a lot to discuss both tomorrow and on Thursday about tumor evolution, and mutual exclusivity. So let's thank the speakers again.